My name is Valérie Marot-Lassauzaie, and I am a PhD student at the Max-Delbrück-Zentrum in Berlin. Today I will be presenting kp-velo, a method to improve single-cell RNA velocity estimation. So first, what is RNA velocity? In single-cell analysis, we are interested in studying dynamical processes where cells change through time. Here we're looking at the example of developing mouse pancreas, where the cells slowly commit to the phosphate of alpha, beta, epsilon, and delta cells. However, because of the destructive nature of the sequencing technology, we cannot follow individual cells through time, but we only get one snapshot for every cell. However, we would still be interested in, even without prior knowledge, finding out the direction of the progression of cells through this dynamical process. And this is where RNA velocity comes in. RNA velocity was first introduced by Lamano et al. In 2018, and is based on the fact that unspliced RNA can be distinguished from older spliced RNA through the presence of introns. So if we follow an RNA molecule through time, it is first transcribed with a rate alpha, giving us a new unspliced RNA molecule, who is then spliced with a rate beta, and this spliced RNA molecule is then degraded with a rate gamma. Now if we plot the unspliced by spliced abundances over time, what happens is as the gene is turned on, in induction phase, first the unspliced counts go up and then slowly the spliced counts follow, until at steady state the amount of each molecule that is being created and degraded are at equilibrium, and then when the gene is turned off again with alpha set to 0, the unspliced counts go down first, followed by the spliced counts, giving us a symmetrical representation of the values as at induction. The RNA velocity is then given as the time derivative of the spliced counts for each cell, and this allows us to predict the future state that the cell would have, and thus the direction of differentiation. Now this concept was first introduced by Lamano et al. In 2018, but it was extended by Bergen et al. In 2020, who recovered the full reaction rate parameters through dynamical fitting. Now the typical RNA velocity analysis workflow starts with the measured spliced and unspliced counts. We have a measurement for every cell, for every gene, of the unspliced abundances, and for the same cell and the same gene, we also have the spliced abundances. And then for one gene at a time, we can recover the alpha, beta, and gamma rates through dynamical fitting of these parameters. After fitting of the reaction rate parameters, we can recover the high-dimensional velocity vector, and then we get a velocity value for every cell, for every gene. Now, because this high-dimensional velocity vector is difficult to conceptualize, we typically want to visualize these velocities on low-dimensional embedding. At the moment, there are still multiple issues with this velocity analysis workflow that we describe and revise in our work. And we will compare our results to current state-of-the-art kc-velo, that is the method that was brought out by Bergen et al. In 2020. The first issue we will focus on is on the recovery of alpha, beta, gamma for every gene. The first issue is that there is a scalar invariance in the solutions alpha, beta, gamma given the unspliced and spliced abundances. We are trying to fit 3 parameters given 2 sets of operations, meaning that these can only be solved up to a constant. In other words, alpha, beta, gamma is a solution, describe these almond shape fit that we see on the left plot, but kappa alpha, kappa gamma, kappa beta, and kappa gamma is also a solution for any possible kappa they describe the exact same almond shape fit that you see on the plot on the left. And now if we look at only a single gene, this is not too much of an issue. But once we start comparing genes, this is becoming more problematic. Let us take the example of two genes for which we are trying to predict their value at the next time step. If we predict the future state of gene 1 to be twice as high as the previous state, and the future state of gene 2 to be twice as high as the previous state, but actually the future state of gene 2 should be 4 times that high, what we predict as a future state is not a value that actually exists in real cells in our process that we try to study. So we have to solve this scale invariance issue to be able to recover the true velocity vector. Okay, so how can we do this? Let's say we have one gene with recovered parameters alpha, beta, gamma, and the real parameters are alpha kappa, beta kappa, and gamma kappa. We have ui and uj, measured counts for 2 cells, and let's say that we also have delta t, a measure of time between these 2 states. We can rearrange the equation of u as a function of t, such that on the left-hand side of the equation, we have kappa delta t, and on the right-hand side, we have only values that we know or that we've recovered by fitting. What this means, in other words, is that if we have this measure delta t, we can solve for the gene-specific kappa. Now, of course, the issue is that we don't actually have delta t, otherwise we would not need to do this velocity analysis. So we need to find a proxy of delta t that we can use to recover the true scaling factors kappa. What we suggest to use in our work as a proxy for delta t is the density of cells, or the number of cells between 2 states. If we have 2 cells progressing through a continuous differentiation process, dynamical process, the number of cells between these 2 states will give us an estimation of the time that is between these 2 states. And here is also shown the example of a slow gene, where you can see that the density of cell over the almond is higher, and a fast gene where this density is lower as the cells are moving faster over the almond. Now, based on this assumption, we can recover the scaling factor kappa on simulation. We simulated a set of genes that follow some true parameters of reaction rate and a true scaling factor kappa, and we see how well we recover this kappa. And the values that we recover are highly correlated to truth. And then we can look at the recovered velocity, the recovered high-dimensional velocity vector, and compare it to the high-dimensional velocity vector that kc-velo recovers. Here we are focusing on 2 genes, a fast and a slow gene, and what we can see in particular for kc-velo is that the velocities for individual genes are highly correlated to truth, but once we look at the relation between 2 genes, we have an issue that is proportional to the scaling factor kappa. In comparison, the velocities for kappa-velo are much closer to truth. Applying kappa-velo on the real dataset of pancreas endocrinogenesis, we can recover a set of fast and slow genes, and what we see is that fast genes are quickly active in part of the trajectory. In the example of NNAT in the middle, it's only upregulated in the beta cells. EYA2, for example, is up and downregulated in parts of the endocrine progenitor cells. In comparison, slow genes are slowly up or downregulated over the entire trajectory. Lastly, when we recover these reaction rate parameters, we can focus on splicing rate, and what we see is that the range of splicing rate returned by kc-velo is in the 10,000-fold. This would mean that if the splicing rate for one gene is 1 second, the splicing rate for another gene would be 2.7 hours. It is difficult to experimentally determine the true range of splicing rates, but none of the reported ranges are close to this. In comparison, the range of splicing rate reported by kappa-velo is 100-fold, which is much closer to the reported ranges from experiments. Now, we've recovered the high-dimensional velocity vector and scaled the genes correctly. the next step is visualizing these velocities. There are multiple aspects that we want to pay attention to when visualizing the velocities. The low-dimensional representation of the velocities should preserve the direction of the vector, but also the magnitude of the vector that represents the speed of change in different regions of the trajectory, as well as local variation. Because we are looking at individual cells, we are also interested in the variation between individual cells that could represent fluctuations of the dynamics and cell plasticities. To illustrate all of these aspects, we designed a simulation where the cells at the beginning of the differentiation trajectory have low velocities and high variance, and then as they commit through fate along this trajectory towards the end, they have high velocities and low variance. And we can see that the PCA projection of the velocity nicely captures all of these aspects that we want to represent. kc-velo's embedding, on the other hand, loses both the information of the magnitude of the vector and smooths over local variation. Now, this is a bit of an unfair comparison, because the reason why kc-velo projects velocities the way they do is to be able to also project velocities onto nonlinear embedding, such as t-SNE or UMAP. To project velocities onto nonlinear embedding, we suggest an alternative method based on the Nystrom projection, and without going too much into detail, what we can see on the right is that the Nystrom projected velocities conserve this information of the variation between the cells, as well as the length of the velocity vector. Lastly, we can visualize the correctly scaled high-dimensional velocity vector with Nystrom projection. And what we see is that there's in the cycling cells, in this ductal population that is still cycling, there's a very high plasticity, and then there's a commitment and speed up in the endocrine progenitor population, who finally commit to these 4 final terminal states of alpha, beta, delta, and epsilon cells. Lastly, I would like to show an example that illustrates the importance of careful processing to prevent the introduction of artifacts. This is a dataset of erythroid maturation, and it was studied by Baril et al. And they found, when running RNA-velocity analysis, that the arrows were predicted to go in the wrong way. And this was not the way that they expected this differentiation to go. And they could trace back these false velocities to a set of Merck genes, so genes that they found had multiple kinetics and were thus incorrectly fitted. And if they removed these Merck genes, they could recover the flow in the way that they expected. But actually what we found is that these Merck genes were in a large part an artifact of processing. In this study, they normalized the spliced and unspliced matrices separately, treating them as separate modalities, when in fact the ratio of spliced to unspliced is very important in velocity analysis and should be conserved even after normalization. If we normalize these 2 matrices jointly, we find that most of these Merck genes don't have multiple kinetics anymore, and that the direction of differentiation is mostly resolved in this erythroid branch. So to summarize, our new method, kp-velo, is able to recover the relative speed of genes and thus scale the high-dimensional velocity vector correctly. We also introduced a new approach to visualize the high-dimensional velocity vector with the Nystrom projection that faithfully represents this high-dimensional velocity vector in a low-dimensional embedding. And we also highlight the importance of careful processing to prevent the introduction of artifacts. If you're interested in finding out more about this work, our preprint is out on bioRxiv. We've also made all the code used in this study available on GitHub, and we are at the moment working on a small Python package to be able to run the whole kp-velo pipeline easily on UData. With that, I would like to thank my group, in particular my group leader, Lale Akverdi, and colleague Bridget Baumann, who worked on this project with me, and as well as everybody else in my group, and you for your attention.